Hello, everybody. Just want to thank the OHBM organizers for giving me the chance to present this data to you. So I'll be speaking on the role of cortical interneurons for in vivo brain function and their potential link for functional correlates of schizophrenia liability. So we know from classic neuroanatomy that brain cell types are not evenly distributed throughout the brain. And this leads to a critical question, which is how brain microstructure influences the function of a given cortical area. And what I mean by this is that certain brain regions, are particularly suited for processing different types of information. For instance, visual cortices are particularly suited for processing rapidly changing visual information. Other regions might be apt at producing ballistic or motor control, and more anterior portions of cortex, prefrontal areas, are suited for processing information over longer timescales and supporting more complex forms of cognition. I'll make the case that cortical interneurons are particularly important for shaping these functional differences across cortex, and I'll show you how we're using or leveraging the genetic data from resources like UK Biobank to make inferences about biology. So, since the interneurons were first discovered, I apologize for my slide clicker here. Researchers have been, there we go, there's the next slide. So since the first discovery of interneurons, researchers have been hypothesizing their particular functional importance for human cognition. So at the end of his career, Ramon y Cajal hypothesized that it was the abundance of these so-called neurons with short axons that was responsible for the complexity and diversity of human brain or human cognition. Now, inhibitory interneurons provide GABAergic mediated mechanisms of inhibition across cortex, and there's a lot of debate about how many subtypes of interneurons there are. For this talk, I'll ask you to remember just two. In particular, these are the two most abundant interneuron classes. The first being somatostatin interneurons, which will always be colored in red. The next being parvalbumin, which will be colored in blue. Now, the reason why we're focusing on these two cell types is because they form stereotyped microcircuits across the cortical sheet. So somatostatin interneurons provide dendritic inhibition in more superficial layers of cortex, as opposed to fast-spiking parvalbumin interneurons, which provide direct recurrent feedback on these excitatory cells. And we have really beautiful computational models from the rodent literature, which states that the relative ratio of these two interneuron subtypes influence the functional dynamics of cortex. So I can't get into the details, but the model hypothesizes that a relative increase of parvalbumin interneurons, my slide will do something at some point, will result in higher amplitude cortical signals, as opposed to a relative increased abundance of somatostatin interneurons will lead to, or is predicted to have, be associated with lower amplitude signals. So I'll tell you three things during this talk. The first is that post-mortem measurements of interneuron distributions throughout the brain closely track in vivo measurements of cortical signal amplitude. Next, I'll make the case using genetics that parvalbumin interneurons in particular underlies variation in cortical signal. And next, or at the end of this talk, I'll show you functional correlates of polygenic risk for schizophrenia and how they implicate parvalbumin interneurons in particular. So my co-speakers have done a lot of the background legwork for introducing the data used in these analyses. Relying on open access data from the Allen Institute for Brain Science. So in short, it provides whole brain gene expression measures. We're analyzing 1,700 samples across the cortical sheet, and we're able to measure the relative expression of individual genes across all of these different brain areas. Also, we are relying on around 9,700 individuals worth of data from the UK Biobank, where we have genetic and resting state data in the same people. So I talked about cortical signal amplitude at the beginning of this talk, and we're operationalizing this with a very simple measure called resting state functional amplitude. It's just the standard deviation of the bold time course. And so more anterior regions, importantly, the signal is not homogeneous across cortex. So in areas of visual, motor, and parietal cortex, we see higher amplitude signatures, which may reflect the fast types of information processing occurring in these areas, which is opposed to more anterior regions, which might be performing more slow evolving or complex forms of cognition. And again, we think that the relative ratio of somatostatin to parvalbumin may be a key evolutionary innovation that allows for this specialization of signals across cortex. So briefly, we summarize our RSFA data using the Schaefer Atlas, and we perform a simple clustering to identify regions of cortex with similar patterns of resting state amplitude. This is mainly to reduce the dimensionality in our data and for visualization purposes, but the take-home of this plot at the bottom left is that regions of motor and visual cortex tend to have similar patterns of cortical signal amplitude that stand in opposition to multimodal or associative regions of cortex. So we then quantify the relative abundance of these two interneuron subtypes using that Allen brain gene expression data. So areas in blue show you where parvalbumin is expressed relatively more than somatostatin. Regions in red show you relatively more abundant somatostatin gene expression in contrast to parvalbumin. And the model predicts that in these parvalbumin-dense areas, we should see high signal amplitude in contrast to high somatostatin areas in these medial, insular, and temporal lobe regions, where we should see low amplitude measures, which is borne out in our data. And this survives proper gene-based permutation tests. And so this tells us that we're on the right track. We've linked ex vivo patterns of interneuron distributions to in vivo measures of cortical brain function. But a stronger and potentially causal test would be to show that genetic variation that influences each one of these cell types underlies cortical signal amplitude in the same individuals. So to do this, we take a guilt by association logic to nominate genes that could be functionally important for parvalbumin and somatostatin cells. Simply, we just identify those genes that are most spatially correlated to each one of these cell types. We then employ a method called partitioned heritability, which, simply put, identifies single nucleotide polymorphisms occurring within the somatostatin gene set, the parvalbumin gene set, and the rest of the genome. And this allows us to partition the amount of genetic variance in a trait and attribute it to each one of these genomic regions. So for any of this to work, we have to show that our primary trait of interest is in some form heritable. And so we find that cortical signal amplitude is between 25 to 35% heritable with about a 10% heritability in visual cortex. We can then ask what proportion of this genetic variance is attributable to the parvalbumin gene set and the somatostatin gene set. And what we find is a notable enrichment of variance explained for these 500 parvalbumin-related genes. So in particular, you should note that the level of variance explained is not uniform across each one of these regions. And I just want to note that the parvalbumin SNPs account for only about 2.2% of the analyzable genome, but are explaining between 6 and 24% of genetic variance in these areas. So we can analyze these data at a much more granular level. So what I'm showing here is the same partition heritability analysis, but conducted for each one of those ROIs. And at the bottom left, those regions where you see increased blue denote areas where this parvalbumin-related gene set is explaining more of the genetic variance in cortical signal amplitude. And you don't have to squint your eyes too much to see that this map of explained heritability is tracking those regions where the parvalbumin gene is expressed the most, and this is borne out in our data when we correlate resting state functional amplitude variance explained to patterns of parvalbumin interneuron marker expression. And again, this survives gene-based correction, but this indicates that the cortical microarchitecture, or the cytoarchitecture of a region, is functionally important for for those types of measures that we are getting from an MR scanner. And I mentioned that I would give you some data about the role of this interneuron type for liability for schizophrenia, and its reduced interneuron activity is one of the core hypothesized pathophysiological mechanisms of the development of psychotic disorders. And so to test whether parvalbumin cells could be functionally important for biomarkers of schizophrenia, we calculated polygenic risk for schizophrenia in each one of the UK Biobank subjects. So regions in blue denote areas where increased polygenic risk is associated with decreased cortical signal amplitude. And if any cell type specific effects could be associated or underlying liability for the disorder, we would expect those effects to be most present in parvalbumin dense areas of cortex, which is borne out in this plot here in the middle. And again, this survives gene-based permutation tests. So to conclude, I've shown you that post-mortem measurements of somatostatin and interneuron distributions closely correlate to cortical signal amplitude, indicating their possible role in this functional phenotype. And I've also shown you that we can leverage genetic information to at least begin to make some kind of cell type specific inference, and that parvalbumin interneurons may be particularly important for bold signal amplitude. Finally, parvalbumin interneurons could be particularly important for functional correlates of schizophrenia liability. So I've skipped a lot of analyses and controls and follow-ups, so if any of this is interesting to you at all, please check out our bioRxiv paper, and all of the code and data will be released as soon as we get out of review. So with that, I'd like to thank my awesome advisor, Avram Holmes, the Holmes Lab, our collaborator, Tien, hey Tien, and all of you for your attention. Thank you.